Okay, so it's 6 p.m. and sorry. Okay, so it's 6 p.m. I'll start the session here. Nobody has joined yet, but I have to start because it's the stipulated time. So everyone who is looking at this recorded live session, hello everyone. I just yeah. Hello everyone. My name is Shomo. I am a second year PhD student at IISc Bangalore, and my work primarily revolves around uh, NGS data analysis, and I primarily work on structural variations, detecting and characterizing the structural variations in the genome. And uh, yeah, let's get this started. Yeah, okay. Yeah, so the course has next generation sequencing technologies, data analysis, and applications. This is week one. The course instructor is Professor Riddhi Nondhar of IIT Kharagpur, and I am the PMRF TA for this course. My name is Shomo Shubhamazumdar, and I am a second year PhD student at IISc Bangalore. So, before we get started, I'll just give you an overview about how these sessions will work. So, what I'll do is I'll try to cover what has been covered in that particular week. So, for this session, I'll cover whatever has been covered by Sir for this week's module, and I'll quickly cover it up. I'll quickly go through the details and the caveats, and after that, in the later part of the session, I'll help you solve some previous assignment questions, and we'll solve some MCQs together. So let's get this started. So okay, so the first topic that has been taught here is basically Sanger sequencing. So when we talk about next generation sequencing, we also should know where this next generation sequencing came from and what was the first type of sequencing that was used to sequence the genome, or at least short stretches of the genome. Because this technology was not so advanced that you can like read the whole genome very quickly. So this was a taking procedure which had been developed in uh, early 1970s, or 1977 to be precise. So this technique was developed by a person called Fred Sanger, who won the Nobel Prize for this discovery in 1977. So before this, we did not have a standardized method to sequence DNA in particular, and so Sanger sequencing was something that made the head start in terms of sequencing different genomes and RNAs and different types of other molecules that we sequence from the cell. So this process was first developed by Fred Sanger, as I said, and is known as Sanger sequencing. And this was one of the main technologies that was used to sequence genomic data until the mid 2000, when this technology was replaced by second generation sequencing technology. So the Human Genome Project that happened in like the late 90s and the early 2000s. So majority of the project was based on using Sanger sequencing to generate reads and trying to get a reference of all of the whole human genome. So Sanger sequencing was the main technology that was used there, because next generation sequencing technologies did not come up at that point of time. So what happens is DNA polymerase naturally uses these deoxynucleotides, which are known as DNTPs. These are triphosphates, and basically these are the units of the DNA. So what happens is DNA polymerase naturally uses these DNTPs that are present in the system. In the biological system, you can call it. You can try to imagine that the DNA polymerase acts when they are trying to replicate the DNA, and inside the nucleus there are these DNTPs, which are recognized by the DNA polymerase, and they're used to synthesize a new strand. So uh, the process occurs very quickly, and makes it very hard to make any sort of measurement during synthesis. That is the issue. So if you are trying to sequence by synthesis, the primary problem is like it will sequence one after the another. You will not know what is getting sequenced. So Sanger overcame this problem by figuring out a way to terminate the synthesis. I thought somebody was coming in. Okay, let's continue. So yeah, so what the issue is, like if you are trying to do sequence DNA strand by synthesis, the synthesis will take after one after another in a serial way, but you will not know what kind of molecule is being what particular nucleotides are being synthesized. So Sanger overcame this problem by figuring out a way to terminate the synthesis using a modified version of DNTPs called DDNTPs. So these are deoxynucleotide triphosphatase. So normal DNTPs has only one deoxyribose. So this has dideoxynucleotide. So it has two deoxy. So two OHs has been converted to H. So it's called DDNTPs. And this DNA polymerase can attach a DDNTP to a sequence just like with normal DNTPs, but cannot attach anything to the DDNTP. So what happens is the DNA polymerase is synthesizing a strand based on a template, and it keeps on adding DNTPs one after the another depending on the complementarity of the strand. But the moment one DDNTP is added, it cannot attach another DNTP after that. So what happens is the sequencing or the synthesis is stopped. So and you can measure and you can see at what position the synthesis has stopped. So in other words, the attachment of a DDNTP halts the replication of the DNA molecule, and this is very similar. Actually, this sequencing by synthesis technology, the main idea of this technology, is still used in next generation sequencing technologies. And uh, like if you compare it to Illumina, uh, both these sequencing technologies, Sanger and Illumina, both use sequencing by synthesis technique. However, Illumina, which is a second generation sequencing, it massively parallelizes Sanger sequencing, resulting in a gain of roughly six orders of magnitude in terms of cost and speed. So, what happened is after Sanger sequencing, we came across different types of next-gen sequencing methods, which basically brings down your cost and massively increases your speed of sequencing. So we'll look into that a little bit later after we see what basically is sequencing by synthesis. As I was saying, it takes the phenomenon of sequencing by synthesis takes advantage of the fact that the DNA strands, which are normally in double helix form, split apart for mitosis and each strand is copied. So Sanger figured out a clever way of converting the sequencing problem into a problem of measuring mass. So DNA polymerase naturally uses these DNTPs to synthesize a new strand, but when you add DDNTP to this, what happens is once a DDNTP is added to the strand, the synthesis stops. So DNA polymerase can attach a DDNTP to the sequence just like DNTPs, but it cannot attach anything to the DDNTP. So basically, once a DDNTP has been added, the synthesis is stopped, and now you can measure the mass of the synthesized strand. In, so this is the generalized overview method of how this sequencing by synthesis occurs. This is not exactly like this is not like only applicable to Sanger sequencing. It is applicable to any kind of sequencing technology that that uses the sequencing by synthesis method to sequence a particular genome. So let's go through the steps. So in the first step on your right, I have showed the steps using a flow chart. So if you follow, and so at first what we do is we first replicate the sequence. If you have a strand of DNA, what you do is you replicate the sequence using a technique called polymerase chain reaction, which is PCR, which is a very commonly used technique nowadays. And what it does is it takes advantage of this DNA replication to exponentially increase the amount of DNA. And what happens is you started with a with a short amount of DNA, and what you did was you tried to amplify this DNA in like same strand of DNA is being amplified multiple times. So what happens is the amount of DNA exponentially increases. So after that, you break down the strands of the two, because DNA is double helix. So you break down both of the strands and using heat or by other methods. And one of these strands can be used for a template strand on which new bases will be attached. So, so you had a short amount of DNA, a small amount of DNA. You tried to amplify it with PCR. After the PCR amplification, you break off the strands of this DNA double helix, and you have the single strands, which are template strands on which new bases will be added on. So you after that you get the template strand. You add this template strand of DNA to a test tube with free floating DNTPs and only a few modified DDNTPs. So here on the left of the slide, you can see the ratio of modified DNTPs, which are DDNTPs. So this T star is showing is denoting the DDNTPs, and the T is denoting DNTPs. So DDNTP by DNTP, the ratio is around 1 is to 99. So basically 1% of your nucleotide phosphatases can be 1% of that can be your modified DDNTPs. So, uh, so as you see, you have a lot of these DNTPs. So TA, GC, these are all DNTPs, and your T stars. It's here it is shown only two. So T star is given as your DDNTPs. Now all the DDNTPs are of the same type. So at one point of time, you only add one type of DDNTP, because if you add multiple types, so the strand will get the strand will stop synthesizing at multiple points of the strand. So at T, it will stop synthesizing when a C, when a modified A comes and binds to that. Again, after a G, it will stop synthesizing when a modified C comes to and binds to that. But what you want is basically to find the positions at which a particular nucleotide is present. So what you do, you only add only one type of DNTP, sorry, DDNTP, and you add the primer or a short sequence that attaches to the beginning of the strand of interest and starts the whole replication process. So if you re recall the replication inside the cell that happens, we call a little bit of molecular biology. So the replication cannot is a process that cannot start. DNA polymerase cannot start the replication process by itself. It requires a small primer that is present at the initial segment of the strand, on which the DNA polymerase will go and bind, and then start synthesizing the strand. So you need a small primer. This small sequence of TATG is what acts as a primer. So you add that so that it, on top of this, DNA polymerase can go and bind and start synthesizing the rest of the strand. So uh, you add the primer and it attaches to the beginning of the strand like this, and it starts the whole replication process. So what will happen is only you have put 1% of this modified DDNTPs. So then for 98 or 99% of the time, it will form the whole strand without any incorporation of the DDNTP. So it'll it started from with TATG, and after that, what it is adding is what it is adding is basically just DNTPs. So because your DNTP concentration is very large. But on the other hand, since you have added those small DNTPs, in small amounts, you'll find these kind of strands. So for example, it started with TATG, sorry, it started with TATG, and then it moved along with the strand C, G, and the next one, the complement to A was T. But instead of adding a T, what it added was a T star, which is basically a DDNTP. So basically this sequencing got stopped at this point. Another another uh, case in which there is another T to be added where there is another A. So at that point also, it can add a DDNTP key at which the sequencing will stop. Now, after you've gotten this modified nucleotide incorporated DNA strands, what you do is you measure the length of each strand with gel electrophoresis to determine at what position has the DNTP gone and binded to it. Say, for example, in this position, the DNTP has gone and bound to it, and in the second case, it has gotten bound to it at a later position. So the mass of this whole strand, for in case of the second strand, will be much greater than the mass of the first strand because already got terminated before reaching to this position. So what in Sanger sequencing, what they do is they run these strands on a gel electrophoresis uh, on a gel electrophoresis plate, and they see how much the DNA travels. So the greater the mass of the DNA, the smaller amount of distance it will travel. So if you can see, this is basically what a gel electrophoresis slide looks like. So uh, the 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 nucleotides that are in top that have lower molecular mass, that they have traveled a lot, but the nucleotides that have uh, the strands that have less amount of DNA, less molecular mass, they have traveled the most, and the strands that have the most molecular mass have traveled the less. So that is how you determine actually what is the weights of these individual strands. So once you do that for one nucleotide, this you did for T, then you repeat it using a modified A, a modified C, and a modified G as well in parallel lanes. So what will happen is at every point a modified DDNTP is added to the sequence, the sequencing will stop. And if you measure the mass, you can have an estimate at what position that sequence has been added or that modified DDNTP has been added. Now, if you know at what position the modified DDNTP has been added, you know the complementary sequence of the strand. So at say this is like 1, 2, 3, 6, 7th position the DDNTP has been added. So what you know is at the 7th position there is A in the original strand. So keep in mind this is your complement strand. So this is not your original strand. Your original strand is this. This is what you're trying to sequence. What you're doing is using complementarity of the DNA, you are trying to find the original strand. So if a DDNTP has been added here, it automatically means that in the complementary strand there is an A. So at every position, this T has to be added. So there will be, like say for example for this one, so we started with the TATG, so these two A's are gone, but the next two A's, this A and this A, need a T to be complementary to each other. So at both the positions, at this A, a modified DDNTP will be added, and at this A also a modified DDNTP will be added. So at every position that uh, that is there, a modified nucleotide will be added and the sequencing will be stopped. So at the position the sequencing has been stopped, you know the position and you know what uh, modified DDNTP has been added. So you can find the the position will be same in your original strand. What you need to find if a DDNTP has been added and sequencing has been stopped, that means in the complementary strand there is an A. Similar is the case with C, G, and A. So this is a process that goes on iteratively, and then until you find all these nucleotides, which are one after the another. So basically, this point you'll need to find a G star, and you can claim that this is a C. This point you'll have to find a C star, where you can claim that is a G. Like this, at every point you want to check where the synthesis is getting ended and what the DNTP has been added to figure out what your complementary stand is. So this is the whole idea of your of basically obviously Sanger sequencing, but the whole idea of what is known as sequencing by synthesis. So after this, as I said, you measure the mass of the isolated strand. This can be done either using radioactive labeled nucleotides and measuring the level of radioactivity, because like if you use a modified radioactive uh, DDNTP or a DNTP here, a radioactive molecule, you can measure the amount of radioactivity coming from that strand, and according to that, you can see what at what position what nucleotide is present in your original strand. So you can do this either by radioactivity, or you can add fluorescent tags to the nucleotides and measuring the strength of the light emitted. So we'll come to this technique. Illumina uses a similar technique like this, but we'll come to that. But this is the basis of sequencing for many types of sequencing technologies. So this was first used during Sanger's time, and after that, this has been developed for Illumina and other technologies as well. So moving on, we understood how Sanger sequencing works, but there are visibly very a large number of limitations and very active limitations of the Sanger sequencing process. That is why nowadays it's not used. So what happens is Sanger sequencing works for sequences below roughly 700 base pair in length. The real limitation stems from the fact that as the length of L of the sequence is increased, distinguishing the mass of a sequence L and a mass of sequence L plus 1 becomes harder. So the longer your DNA is, the harder it is to find after the end of that uh, of a nucleotide, what is the next nucleotide. So for example, your L is very large, it is very hard to distinguish a DNA of length L with a mass M with the DNA of length L plus 1 with a mass say M star, because M and M star will be very similar. So the larger your DNA fragment is, the more harder it is to resolve them according to the length, according to the mass. So for larger sequences, Sanger sequencing does not work well. And to see this, note that a tolerance of 0.1% in measurement would make it impossible to distinguish a sequence of length 1000 from one of length 1001, even if all the bases had the same molecular weight. So it is very hard after a certain amount of length to sequence the DNA using Sanger technique. And such errors in measuring mass are also for reasons are also a reason for errors in Sanger sequencing. So ultimately, you're basing your uh, logic in the fact that like you need to distinguish these small fragments of DNA based on their mass, but it is not always possible because, like as I said, as the length increases, it becomes more difficult to resolve the two different sequences of DNA. But what happens also is, like, during this distinguishing, there are also you ultimately you are measuring the mass and comparing across different stands. So here also there is a point of uh, error where uh, point where an error can be introduced into the sequencing. So measuring mass is it's not a very easy thing to do, and it's like if you even do it objectively, there will be a lot of errors. So this is one of the reasons why Sanger sequencing we see a lot of errors in Sanger sequencing. So this was a sequencing we talked about Sanger sequencing. So this was a sequencing that was used in uh, the late 1970s and started at that point of time, and it was used till like mid of 2000s. So after that, we saw a very uh, exponential rise of sequencing technologies, bringing down the costs and also bringing down the time as well, time to sequence as well. So one such technology uh, before next generation technology was using microarrays. So microarray is not exactly sequencing. To uh, just to remind you that microarray technologies do not kind of basically sequence the whole genome. So what happens is this microarray are solid supports, usually made of this glass or silicon slabs, upon which a DNA is attached and in an organized, predetermined grid fashion. So you have a slide like this, you attach this DNA probes at different points in the slide, and you know where which probe of the DNA has been attached. So each spot of the DNA is called a probe, and it usually represents a single gene. And these DNA microarrays can analyze the expressions of tens and thousands of genes simultaneously. So you have a like kind of complementary strand of DNA molecule which is complementary to your gene, so that if the gene is getting expressed in your cell, and after you culture the cell and you isolate the RNA or a DNA, and this particular strand will come and bind to the strand on your microarray grid, because the strand on your microarray grid is already complementary to the strand your strand of the gene that you're getting from the cell. So you take different cell types, you culture them in the lab, and you isolate RNA or DNA, whatever you're trying to measure. And then what you do is you do reverse transcription. For RNA, you do reverse transcription. For DNA, you do replication. And you add these fluorescent tags like this at the end of the strands. So what happens is these strands go and bind to this DNA probes and emit this kind of light. So I'll not talk much in detail about microarray technologies, but you need to know the basics of how this technology is used. Because even now, if you go to certain databases, say for example, gene expression of omnibus, which is GEO, you go there and find transcriptome databases. You'll find a lot of data. Uh, you'll find a lot of data sets that have studied RNA expression, basically the gene expression, using microarray technologies. So this is a technology that has been all widely used, but now because we have better technologies, we are slowly moving away from microarray. But still, it is a very cost-effective and a good method to find in targeted part of your genome and see if there are any mutations in there or not. So one advantage of DNA microarrays is it can analyze the expressions of tens of thousands of genes simultaneously. So parallelly, you can do you can analyze the expression of these genes, and there are many synonyms for DNA microarrays. Sometimes they're called DNA chips or gene chips or DNA arrays or gene arrays or biochips. All are the same thing. It's basically you have a plate on which different probes are attached. According to that, if you extract all the genetic content of your cell and put this inside the plate, the strands that are complementary to this DNA probe will come and bind, and since you have attached it with a fluorescent light, it will give you a signal. So, for example, here is a difference in different kind of signals. So, for a healthy cell, you tag it with basically different kind of a signal compared to a pathological cell, and you can check where the at which points there are mutations inside in that particular strand of DNA. So this is how the output looks like. What if you see from on the top? So you find these different colors emitting from the probes because you have attached with the fluorescent tags. And after that, you can analyze what exactly is basically what exactly is the issue with your pathological cell compared to a healthy cell. So, the principle of DNA microarray technique lies on the hybridization between the nucleic acid strands. So, that is the most important part because you already have a DNA probe. You need another DNA to come and bind to it and release a signal that you can capture. The property of complementary nucleic acid sequences is to specifically pair with each other by forming hydrogen bonds between the complementary nucleotide base pairs. For this, samples are labeled with fluorescent dyes, as I was saying. At least two samples are hybridized to the chip, and complementary nucleic acid sequences between the sample and the probe attached on the chip get paired via hydrogen bonds. It's easy, like normally how DNA forms a double strand. I guess all of you know it's using hydrogen bonds. So complementary nucleic acid will come and kind of bind on your DNA probe, and because they are complementary, they'll form these hydrogen bonds and become double stranded. The non-specific bonding sequences which remain unattached and washed out during the and the and are washed out during the washing step of the process. So after this is bound, you go through a washing step where basically these non-specific bonding sequences will uh, will be washed out, and the fluorescent label target sequences that bind to a probe sequence generate a signal, as I was saying. The signal depends on the hybridization conditions, for example, temperature. Also on how you are washing after hybridization. So there are certain factors on which this signal depends on. And it also depends upon the amount of the target sample that is present. So if only say there is only one strand of the nucleic acid that is you are targeting, and if you have not amplified it enough, so you will not get a signal, because it's very unlikely that there is only one strand present and that strand will find the complementary strand on your DNA probe and go and bind to it. It is very unlikely. So you need to have a decent amount of sample present, or you need to amplify your samples so that you can get a better signal. And using this technology, the presence of one genomic or cDNA sequence, and this is basically lakh or more sequences can be screened in a single hybridization. So it's a fairly big number. It's like 1 lakh or more sequences. So uh, this technology is useful, and it is still used till date to measure gene expression and other stuff. But this is a uh, very useful method that, and it's also like not very costly. So this is one of the very useful methods that is still in use, but we have better technologies now. So so coming to the advantages and disadvantages of this microarrays. So advantage is basically one you can think of very intuitively, is that it provides data for thousands of genes in real time, and it can basically uh, give you the amount of tell you the amount of expression of, like, say 20,000 genes. 20,000 is too much because you need to have a very big microarray to get that. But even like say 10,000 genes, if it is giving, or 5,000 genes, it can give you the expression of those genes in a parallel fashion. Like, you do not need to process one gene, then you go and do another gene, and then another gene. You don't need to do that. If you design your microarray in such a way that it can have those 10,000 target sequences on your on your microarrays probes, you can easily uh, parallelize this process and get the expression of these multiple genes together. So it's useful in that manner. And as a result, in a single experiment, it generates many results very easily, and it's fast and it's easy to obtain results, and it's promising for discovering cures to diseases and cancer. And different parts of DNA can be used to study gene expression, so you have a lot of flexibility here, a lot of, what to say, depending on your requirement, you can design your own DNA microarrays and check expressions for the genes that you like. But one thing is, it's that there are also disadvantages, so that this technology is getting replaced by newer ones. First thing is, it is the creation of this DNA microarray, depending upon your requirements, it's a little bit expensive to create. And the production of too many results at a time requires long time for analysis, which is also quite complex in nature. And these DNA chips also do not have very high shelf life, so they are not that usable. So you have to make kind of like, you can assume you have to make DNA microarrays for each single of your experiment. It's not that you make one microarray and you can use it for all your PhD life or 5 years. It's not possible because they have very short shelf life. So it's a little bit expensive on that front because you need to make it every time. So yeah, but it is very fast and it's kind of well reliable as well. So yeah, I'll strongly suggest you go to some transcriptomic databases like Gene Expression Omnibus (GEO), and you'll find the datasets. A lot of datasets are there measuring gene expression of particular samples in control or in disease states. They have all been done using uh, microarray technologies. After microarray, if you want to measure gene expression nowadays, the technique is to use bulk RNA sequencing. Also, there are more sophisticated techniques now where you can measure this amount of expression from a single cell. So that is single cell RNA sequencing. But yeah, technologies have evolved, but this is still quite a useful uh, technology that is still used till date. Okay, now we come to the main part, which is the next generation sequencing. So, what is it? NGS or next generation sequencing is a robust platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. So, this is the state of the art right now, but like this, it is still evolving. So, the next generation sequencing, also known as high throughput sequencing, is the catch-all term to describe a number of different modern sequencing technologies. The high demand for low-cost sequencing technologies have driven the development of high-throughput sequencing, which basically what is the meaning of high throughput is basically producing thousands of millions of sequences at once. So it's massively parallelized and it's like you can get millions of sequence at one point of time. So it's very effective and they are intended to lower the cost of DNA sequencing beyond what it is possible with standard dye terminator methods. So, as I was speaking, the dye terminator is in like you add DDNTPs or other type of molecules and you attach them with different kind of dyes or fluorescent molecules, and you can check the sequence from there. But NGS is much more reliable than uh, uh, NGS is much more reliable and basically lower costing as well compared to the standard dye terminator methods. Thus, recent technologies has allowed us to sequence DNA and RNA more cheaply and more quickly than the previous used Sanger sequencing, and such has revolutionized the study of genomics and molecular biology. Classified into different generations, NGS uh, has led to overcoming the limitations of conventional DNA sequencing methods and has found a usage in wide range of molecular biology applications, as you will see in the later part of the course. So how you can detect diseases, how you can detect different kind of mutations which can be resulting to the disease. So you can do a lot with genomic data right now, and that is all due to NGS because at such a short amount of time, so much data you can generate and different types of data also you can generate which you can study and analyze and uh, have molecular biology applications depending on your findings. So okay, moving on. So this is a general workflow for next generation sequencing. I'll just check once. Somebody is trying. Okay, yeah, I'll just check if okay, yeah, okay. So this is a general workflow about of how next generation sequencing works. So for the major four steps. So first is the DNA extraction. You extract the DNA from the cell, and then you prepare the library. So you fragment this DNA whole DNA molecule. You fragment it into smaller pieces. Then you attach adapter molecules on both sides of these stretches of DNA, and then what you make is called a DNA library. And then is the main and the important part is sequencing part. You sequence this libraries and try to get the whole sequence of the DNA that you have given it. And the last and not the least is one of the most important steps is basically analysis. So you have the reads and have the information, and how do you use this information to find different kinds of mutations, different kinds of variants, different kinds of structural variants. There can be big variants, there can be small point mutations, anything can be present in the DNA. So how do you identify those, how do you get those, and how do you use that information uh, to have better diagnosis and have better treatments for particular diseases or particular conditions. So it is very useful in that manner. And uh, till now, I only have talked about the sequencing part. I'll not talk about the analysis part in today's class, but in the future classes we'll talk about it. But this analysis part has also been uh, growing at a very fast pace. So as we know, sequencing technologies have improved a lot from Sanger sequencing to what we have today in terms of long read and Oxford nanopore and whatnot. But uh, along with the development in sequencing technologies, we have seen a massive development in computational power. So we have a lot of computation power right now. So we can generate a lot of data and we can analyze it uh, very well because of the amount of computation power that we have that has been developed till now. So along with development of the sequencing technologies, development of computational power and better bioinformatics algorithms and better uh, uh, analysis techniques is what goes hand in hand. So not increasing the throughput will not only not help only if increasing the throughput will only help if you have a good infrastructure for handling that amount of data and like analyzing that amount of data. So the both is important. Sequencing technologies need to get better, and also in algorithms and computation of analyzing this data also needs to get better. So yeah, this is the general workflow. Uh, yeah, today we are mostly talking about sequencing. In the further classes, we'll talk about the analysis part. Okay, so majorly, like you can have three types of sequencing. Like this is, I'm not talking about in terms of the technology, but in terms of what you're trying to get out of the data you're trying to get out of the sequencing platform. So one is basically whole genome sequencing. So you like you try to sequence the genome from end to end. Another type of sequencing will be exome sequencing. So you do not maybe do not want the whole genome, but you want specific parts of the genome, specific parts including the parts of the genome that are making the genes, that are making the proteins. So that will be the exome part of the genome. Because as if you remember, the in eukaryotes, in humans, so the gene comprises of exons and introns, one after another, depending on the gene, and then the introns after basically getting the RNA. After the RNA is being synthesized, the whole RNA is spliced off and the introns are taken out and the exons are joined together, and after that the uh, mRNA that go into making proteins. So say for example, if there is a mutation in your genome which basically is modifying a particular protein which is contributing to a disease. So what you want is basically what mutation is present in the genome which ultimately is hampering this protein. So you what you try is basically only to get the part of the genome that is contributing to making of the protein, which will be the exome. So you get all the exomes together and you try to find where the mutation is, if there is a particular variant that is contributing to the malfunctioning of the protein or the system. But uh, keep one thing in mind, like the amount of exome in a human genome is very low. It's like less. It's close to 1% of the uh, uh, it's close to 1% of the whole gene. So it's the amount of DNA that actually codes for a protein is very low. So actually exome sequencing is a very cost effective method to find out variants that are present only in the gene, inside your genomic sequences, inside your exons of those genes. So exome sequencing becomes a very good uh, method for that. Also, since you have to sequence very less amount of DNA, because exons are, as I said, 1% of the DNA, so it becomes cost effective as well. Sure, you lose out on a lot of data. You do not know how the introns are looking like, if there are any mutations in the introns that are kind of like contributing to this malfunction of the protein. You won't know that. But if there is a particular mutation or a particular variant inside the exome of the of the of the genome of the individual, you can predict due to that mutation how the different proteins are getting changed downstream and how it is contributing to a disease state. So this is also a very useful method, although like it takes up like it's a little bit cost effective and little bit you can call it as um, uh, a makeshift method, but it's actually very useful. And since it is cost effective, it becomes much more useful than it looks like. So, okay, so this was genome sequencing. We talked about exome sequencing, and another is targeted gene panel. So, what you want is particular regions of the gene that are getting that you think are getting mutated, that you think have issues. So, what you do is you try to target a particular uh, portion of the gene and sequence that particular portion. Hi, I I think you're the first person joining this session. So I have covered it till a little bit, but you can continue to listen from here on, and I'll post the whole video on YouTube. So if you want, you can go and take a look later. But yeah, please bear with me. Okay. Okay. Please ask if you have any questions. I'll just move in from here. Okay. Uh, So now we come to what is known as pyrosequencing. So it uses the Roche 454 sequencing method. And what pyrosequencing was, it's basically developed by 454 Life Sciences, and later it was acquired by Roche Diagnosis. Uh, what it does is it basically amplifies DNA inside water droplet molecules in all solution, which is basically emulsion PCR, and with each droplet containing a single DNA template attached to a single primer coated bead that forms a clonal colony. And the sequencing machine contains picoliter volume wells, so it's a very small volume wells, and they contain this single bead and sequencing enzyme. So you have the bead, you have a well inside meaning you have a well inside that you have the bead, and you also have a set of sequencing enzymes. And the spider sequencing uses an enzyme called luciferase, which basically generates light, and this light is detected, and this is detected for individual nucleotides added to the nascent DNA, and the combined data, the amount of light and what light is getting generated, you can use that data to kind of combine the whole sequence and get a sequence readout. So this is the overview of how it happens. So, say for example, this is a strand you're trying to sequence. So this is your DNA polymerase, and you are in your solution you have DTTP. So basically, it's you add the T, which has um, basically uh, TTP. You add it there, which removes releases a PPI. It's basically a biphosphate. Hello. Yeah. It's basically a PPI, so which basically is a phosphate which binds to AMP. As you know, AMP with phosphate, what it gives you is an ATP. Now, this ATP will react with the enzyme which is present inside that well, which is known as D-luciferin, and it makes the luciferase luciferin AMP complex and releases oxygen. Now, this complex is a little bit unstable, so what happens is it breaks down to give luciferase and oxyluciferin along with AMP, carbon dioxide, and this light. This light generated is what is a marker of what the base pair has been added. So you have these peaks of these lights at different sequence intervals. So you know at this peak a T has been added here. So what it uses is basically the function and the property of the luciferase enzyme, which basically is getting formed due to the ATP that is getting formed due to this phosphate, and this ATP is binding to D-luciferin, which the complex is very unstable, which is breaking down to give you that luciferase and the light which is getting detected by your whatever mechanism you have kept to detect that light. So at every peak, you know what uh, nucleotide has been added to it, and so you can combine all this one after another and you can get a whole sequence. So this is a technology that has since been, like, after 2007 or 8, this is no longer in use, but this is one of the earlier sequencing technology, earlier NGS technologies that was quite famous at that point of time. Okay, so now we come to the the like the most common of sequencing technologies that is used in today's date is basically Illumina sequencing. So this is what is the second generation of sequencing, and this was pioneered by Illumina, and it makes a little bit little modifications to the Sanger process. As we talked about earlier, Sanger process uses sequencing by synthesis to generate the sequence. Illumina actually uses that technology, but in a much more defined manner, much more technologically advanced manner. So the sequencing procedure is massively parallelized in case of Illumina, and this dramatically increases the throughput while decreasing the price. So I'll just go over this. Sorry. Yeah, I'll just go over the method about how it goes on. So first, it Illumina the major advantage of Illumina is parallelization. You can synthesize multiple strands at one point of time. So Illumina achieves this parallelization by running several synthesis experiments at once. As I was talking about sequencing by synthesis, what Illumina does is it parallelizes the sequencing by synthesis. Like so, for each strand, you do not need to wait for one strand to end to get to another strand. So it parallelizes that problem. So what you have is sequencing by synthesis, but across different number of strands. So for in Sanger, you could do it only for one strand at a time. In Illumina, that number has been massively parallelized. So each of the many template strands is anchored on a chip, and only DDNTPs with fluorescent tags are available during the synthesis procedure. No DNTPs are there. So any uh, DDNTP will come and tag to the come and attached to the strand using the DNA polymerase. That will be the end of that strand, because after a DDNTP has been added, you cannot add anything else. So each type of DDNTP is tagged such that it emits a different wavelength or color, and depending on this wavelength of color, you identify what DDNTP has been added. And this, since DDNTP halt the synthesis of the new strands are synced. Sorry. Yeah. Uh, all the strands are of the same length, and at the end of each synthesis cycle, at which a point at which point a picture of the chip is taken. So I'll show you how the picture looks like, but bear with me till then. So yeah, the idea is you have this tagged DDNTPs inside your solution. So you have a DNA polymerase and your strand. So what will happen is this DDNTPs will go and bind to the complementary strand, what is given inside this, or what strand you have provided, and just after the DDNTP has been added, the sequencing will stop because you cannot add more bases after the DDNTP. And after the DDNTP has been added, it will also emit a certain wavelength of signal by which you can identify what DDNTP exactly has been added. Is it a T? Is it a modified A? Is it a whatever it has been added? So, and these pictures, this light emitting pictures are taken by the machine, and these pictures are then analyzed by something called a base caller software, which identifies or calls the complementary nucleotides. And base calling will be discussed a little bit more in the next lectures, but yeah, basically what happens is you take the picture, there is a software which is the base caller, and we identifies at which point what base is there. Sorry. Okay. Okay. So, to override the chain termination, Illumina uses reversible termination. The sequencing process introduces an enzyme which can turn a DDNTP into a regular DNTP after it has bound, allowing the synthesis reaction to continue instead of being permanently halted. So what happens is, once a DDNTP has been attached, it takes a picture to check what DDNTP has been attached. After that is done, what it does is it uses reversible termination. So what happens? It converts the DDNTP to a DNTP so that more bases can be added after that. So once a DDNTP is added, if you do not modify that, no bases can be added further to that. But what Illumina does is it converts after it has recognized what DDNTP has been added, it has recorded what base is present in that position. It it converts this DDNTP into a regular DNTP, which can be after which can be another DDNTP can come and bound and can take a picture. So it's an iterative process. So you start with a strand, then you add a DDNTP, it emits a light, you take a picture from above, you know at that position this is the base. After you know that, you convert this DDNTP to a DNTP, and then you add another DDNTP. And like so, at position A, if you know at position 1, if you know it has A, after you know it has A, you convert the DDNTP to the DNTP, so you can see what the position or what the base is at the next position. So for HiSeq, which is a Illumina sequencer, a single cycle of adding and capturing takes about 6 minutes. So it's actually quite fast. So this is how uh, the basically the picturing looks like. So if you have this DDNTP here, like a green DDNTP has been added, here another yellow DDNTP has been added. So for in cycle 1, what will picture is a green light emitting and a yellow light emitting. The next step, what happens is, this is a DDNTP. Now this will be converted into a DNTP, and the next base will be added here next to it. Sorry, next to it. So after the green, a red has been added. Here also after the yellow, a red has been added. So it's emitting these red lights. So this cycle 2 captures these red lights. Sorry. So at each cycle, a new DDNTP is added. It emits a light depending on what fluorescent things you have tagged it with, and you capture this light using the machine. And there's a software in the machine that can see this light and give a prediction that which base is that at present at that point. So in cycle 1, green and yellow has been added, so you get a green and yellow light. Now after this green and yellow has been added, these are DDNTPs. These will be converted into DNTPs so that the next base can be added. So the next base is added here, both are red, so you get a picture like this. Again, these are DDNTPs, you convert them to DNTPs again, and then the next base gets added. So one by one, you can control one after the another how bases are getting added, and each pointer base a DDNTP is getting added to it. You are getting a picture, and you know what base is getting added to that. So like this, you synthesize the whole strand. So I hope I have explained to you how, the way I can, but this video is basically the official video that is present in the Illumina YouTube channel. Also, you can go and check. I'll put this link in the chat box as well. But this explains the Illumina sequencing very well. So please look at this video.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different
Someone is trying to enter.
ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments.
These adapters, remember, these adapters are very important. We use these adapters as identifiers of these strands. So the adapters you are adding in these steps is very useful at the later stages when you are trying to get from where this sequence has come. These adapters will help you to know, because you have already tagged it earlier, so you have an information what adapter is been tagged to what sequence.
Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the.
This flow cell and this massively like the small lanes inside the flow cell is what gives the power to parallelize this whole process, to parallelize the whole sequencing process. So this flow cells are very important. Just a second. Sorry.
Surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read.
Sorry guys, somebody is trying to join the meeting.
With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only
Just remember, these are DDNTPs. Okay, so these are DDNTPs tagged with these fluorescent tags. So once a DDNTP goes and binds, it will emit this light, the tagged light, that is fluorescent tag that it is with, and the machine will capture this light that is coming out. So it will know that at that position it is this base present. So for A, in this case, it is given as blue. So if the machine sees blue, there is a base caller software inside the machine which will identify that the blue light has come. So that means at that position there is an A that is getting attached. So the complementary of the A will be T. So your original strand, there is a T, and during your synthesis an A is getting attached to it and emitting the blue light. That is how you are getting to know at that point an A is getting attached. Now after a DDNTP has been attached to that, you need to convert the DDNTP to a DNTP to further this process. So after this A comes and joins here, so what will happen if this is a DDNTP is that the synthesis will stop. But you don't want that. You want the whole sequence. So after this has bound and you have captured what light it is emitting and you have known what base is there, after that you convert this DDNTP to a DNTP so that the next DDNTP can go and bind. So this is a very controlled manner in which you are trying to see what bases are getting attached after what, and you can capture that whole information in using the base caller. So yeah.
one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell.
This is what the base caller sees. So this is what the machine captures, and this is an image data which is analyzed by a base caller software, which gives it, which provides at which point and emitting which light is associated with each base. So your base caller does this conversion of information from this image to your sequence. So yeah, just to remember that.
After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are...
So what you're essentially doing is, just to help you understand this intuitively, what you're essentially doing is you're reading the strand from both ends. So you have amplified the strands and you have cleaved the strands and made two different strands. One is a forward strand, one is a backward strand. So what you do is, at one point of time you are reading the sequence from the forward to the backward manner, and in the other strand you are reading the sequence from the backward to the forward manner. So you're reading the same strand in two ways. So this is read 1 and this is read 2, forward and reverse reads.
repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation.
Exactly. Now you remember how that adapter and this sequence of small index that is you are incorporating it in the first step, that is important, because here when you come to the analysis part of these reads, this is where you can use those indices to identify where basically which part of the genome in your original genome the reads came from. So yeah.
For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification.
Okay, I think that is all. The variant identification and the alignment and everything, will come to that in later classes. Just wanted to give you an idea about how Illumina sequencing works. So the idea is quite simple, but it's very effective and it's very useful. Illumina is one of the most used sequencing technologies in our current time. So the idea is that you have the PDNTP, you add to the sequence, it generates a light, you capture the light, and you try to see from what nucleotide that light has coming, and you predict that at that position there is a base that is A or T or G or C, whatever, depending upon the light that is getting captured. One thing to keep in mind, the base caller software that identifies this light and assigns a base to that particular position of the genome is the output that it gives is basically a probabilistic value. So it will give you a probabilistic value between 0 to 1, and depending on that value you can be confident if
Hello.
that value is, yeah.
Yes. बोलिए मेरे नाम से
Sorry, can you mute yourselves, please?
ये आप आ जाइए ये पैट मैट्रिक्स है ना पैट मैट्रिक्स है हाँ क्या
Okay. Yeah. So the output of the base caller is basically a probabilistic value at along with the base that is present there. So it will give, like, at that position maybe there is a base A with this percent probability, say for 95% probability. So it's all based on probabilistic values, because you do not know exactly what the base there is. You are identifying the base using a proxy of this light that is getting emitted when that nucleotide is getting added to the sequence. So yeah, it's a very interesting and a very useful process that we have in Illumina's short read sequencing. Okay, moving on. Not all is good for Illumina sequencing. There are a lot of limitations. So after Illumina, a lot of sequencing technologies have been developed. Now we have very long read sequencing and stuff. But yeah, Illumina has its limitations. So the major limitation that Illumina has is the size of the fragments that can be sequenced. It's around that 150 base pair mark. So in some cases it can be a little bit bigger, some cases it can be a little bit smaller. But if you think about 150 base pairs in the context of the whole genome, our human genome is like 3 billion base pairs long. Billion is like 10 to the power 9. So in among 3 billion total reads, a 150 base pair read is very small. It's very small. It's nothing compared to a 3 billion long genome. So what happens is using these small reads, it is very hard to identify the variants, the big variants that are present in the in the genome. Say for example, you have a duplication inside a genome which spans say uh, 5,000 base pairs or even more. So these kind of duplications that you see in the genome, that can be very like that cannot be very easily identified using Illumina sequencing, because Illumina read length is around 150 base pairs. And it's ideally it's not more than 300, but the average is around 150. So it the more bigger drastic changes in the genome, it becomes harder to identify using short read technologies like Illumina. So, uh, and this limitation is due to the chemistry used in the sequencing process, which relies on short stretches of DNA being attached to the glass plate. These larger fragments of, if the fragments of DNA are larger, they cannot be attached as easily, and therefore they cannot be sequenced as accurately. Illumina might not be an ideal option if getting large DNA reads is crucial for your experiment. So, as I was saying, if you're trying to identify very big variations inside the genome that spans about like thousands of base pairs, Illumina maybe is not the right way to do that, because the read length is so small, you'll have very little confidence in the fact that you have discovered that large stretch of DNA. It will be mostly false positives, and you'll have no confidence in the fact that that duplication or the insertion or the deletion is present inside the genome. So for those big variations, which we call structural variations, anything above than 50, more than 50 base pairs. So for structural variations, Illumina is not the ideal technology to use. There you use more recent technologies where you have longer reads, say for 1 KB or even more. Like even we have sequencers currently available which can give you around 1 MB of read length. So those are the ideal uh, read ideal sequencing platforms or sequencing technologies you should use to identify those big variations. So another and the major limitation of Illumina is there is no real-time data access. You have to wait until the whole process is over, and then only after you can begin the analysis. So the newer technologies has this real-time data access that you can see, and if you can have the real-time access, you can also check if there are certain errors going on with your experiment or not. So this becomes much useful in case of other technologies. Okay, so moving on from Illumina, the next big sequencing technology is SMRT. One second. Yeah. So SMRT sequencing is also based on sequencing by synthesis approach, and the DNA is synthesized in a so-called zero mode wave guides. These are ZM. This is called ZMWs, which are basically these are small well containers with the capturing tools located at the bottom of the well. So you have a plate like this. You have small wells inside the plate, and at the bottom of the well, there is a there is a mechanism by which you can capture whatever it is, be it a light, be it a radioactivity, whatever you're trying to capture, that machine is present at the bottom of the well. And these small containers are called what are known as zero mode waveguides. So the sequencing is performed with the use of unmodified polymerase and fluorescently labeled nucleotides flowing freely in solution. See, the whole idea of sequencing by synthesis, that was same in Sanger, that was same in Illumina, that is kind of same in SMRT. But what they have done is they've modified it according to the convenience and like how you can increase the number of reads, how you can parallelize this process. In those cases, they have improved, but the whole idea remains the same. It's kind of you have a modified uh, DDTP, DDNTP, or NTP which basically will go and bind to your existing strand and it will emit a signal, be it a light, be it radioactivity, whatever it is. The idea is it will go, the modified base will go and bind to your current strand, and you'll know at that position, depending on what it is emitting, it is if it is light, if it is radioactivity, depending on the signature of that signal, you can identify at that position what piece is present. Okay, so the wells are constructed in such a way that fluorescence occurring by the bottom of the well is detected. So in this case, it is detecting fluorescence. You can also detect radioactivity as well. So the fluorescent level label is detached from the nucleotide at its incorporation into the DNA strand, leaving an unmodified DNA. And this can go up to, like, you can get reads up to like 1,000 nucleotides, which is longer than what we saw in Illumina, which was an average of 150. So this is a general overview of about how SMRT works. So this green thing is your enzyme, which is your DNA polymerase. This is your original strand, and this is what you're sequencing. Uh, this is basically the strand that you're sequencing to identify what is there in your original strand. So inside the well, what happens is fluorescent tagged nucleotides are there, and using this DNA polymerase, it attaches to the strand one after one other. So inside the well, here is the detection zone, basically, which will detect the signal emitted after addition of a single nucleotide, and it can tell what nucleotide is being added at that position. So this is your template, the thing you're trying to sequence, the sequence you're trying to know, basically, and this is your sequence fragment, the one that you're sequencing with your modified DNT piece. So what will happen is it will emit a fluorescent signal upon this nucleotide addition, and the signal from each complementary nucleotide is read as it is added to the sequenced fragment. So the idea is quite simple. You have it's basically sequencing by synthesis. Inside the well, you have a detector at the bottom. So after each of the nucleotide has been added, it can detect what nucleotide is being added at that particular time. So one after another, it can tell you what bases are being added to this original template strand. So this the sequence that you get from basically the sequence fragment that you are getting, the sequence of the sequence fragment, the dead fragment, is basically complementary to your original strand. Keep that in mind. So the information that you're getting, what you need to do is find the complementary of those strands, because the original strands is basically complementary to what you are synthesizing and what you are capturing. So the origin, you need to keep that in mind. At the end, the original strand will always be complementary to the strand that you are synthesizing, because that is how DNA polymerase and replication works. Okay, so here there is a concept of hi-fi reads. This SMRT is basically a PacBio technology. PacBio Life Sciences is a company which developed. So, for example, in the earlier Illumina sequencing, it's Illumina is the company that developed that sequencing technology. In this case, SMRT was developed by PacBio. So they have a concept of what are known as hi-fi reads. So hi-fi reads, what are they? These are reads that are produced using circular consensus sequencing mode on PacBio long read systems. Hi-Fi reads provide base level resolution with a very high accuracy, excuse me, using single molecule read accuracy. And Hi-Fi reads can be used across a range of SMRT sequencing applications, from whole genome sequencing for de novo assembly, comprehensive variant detection, anything you want to do with the sequence data. So the idea is you start with a high quality double stranded DNA. This make this circular basically circular double strands on both ends, which basically helps you build the library of the SMRT sequencing process. So as you remember from Illumina, sorry, from, so before I started talking about this, we talked about library preparation, how you fragment the DNAs and you add adapter sequences on two ends to get this library. So this library preparation is a slightly bit different in case of in case of SMRT and Hi-Fi reads. So you have this circular DNA fragments at both ends, and what you do is you anneal primers and bind DNA polymerase, and you kind of use a double stranded circular position. So the circular DNA is synthesized in repeated phases, small repeats like this. It is synthesized like that, and the polymerase reads are trimmed of adapters to yield subreads. So after this is done, this is trimmed, and what you get is basically consensus and the methylation status, and which are called from the subreads. So depending on how this circular DNA fragments are getting synthesized and how these are getting basically how that part of the DNA is getting replicated, you can get what is known as a hi-fi read, which basically is a very highly consensus sequence of the read that you are trying to sequence. So just keep in mind, this is just a technology that is kind of improving your read quality. So what it is giving you is a read that is very highly consensus with what you are trying to sequence. So what happens is, in some cases, the sequence, obviously sequencing is not a perfect sequencing or synthesizing this DNA fragments or DNA strands is not a perfect process. There will be errors associated with it. But if you follow like kind of a multiple times using circular DNA, if you try to replicate that DNA fragment, what you'll get is a read with very high accuracy. So that is what hi-fi reads are used in PacBio. This is just a uh, technology to make your read accuracy better, but this is not the whole story. Basically, that is what I'm trying to say. So after SMRT, basically I should have covered this before SMRT. This is not a better strategy than SMRT, but this is what is known as ion torrent sequencing. Sir has also covered this. So the idea is, when a complementary nucleotide is incorporated to a strand of DNA, a phosphodiester bond is formed, which releases an H plus ion inside the medium, which changes the pH of the medium. And this change of pH is basically sensed by this ion sensor, which is present inside the medium, and it converts this chemical information, this change of pH, how much pH is getting changed, uh, into which is the digital electronic information, is basically what read is present there. So after addition of each read, the amount of H plus that is getting released and the amount of pH that is getting changed, that is very specific to each kind of nucleotide. So if a sensor senses that it has, say for example, I'm just giving a rough example. If a sensor senses that the pH has been increased by 1, maybe an A has been added. If the pH has been decreased by 1, maybe uh, a T has been added, whatever be it. There's a signature associated with the addition of a nucleotide to a strand, which is basically change of pH, which an ion sensor senses and converts this chemical information into a digital information. And as in other kinds of NGS, the input DNA or RNA is fragmented. It is around 200 base pairs, and adapters are added, and one molecule is placed on a bead. These molecules are amplified on the bead by emulsion PCR, and each bead is placed on the side, on the single well of a slide. So, like the Roche sequencing, as we were discussing, uh, this well is basically flooded with single species of DNTP, along with different enzymes that are required to synthesize the DNA on that strand. So that will include polymerases, that will include buffers and everything, and it can add one NTP at a time. The pH detected in each of the wells, as well as the H plus ions released, will decrease the pH. This change in pH allow us to determine if that base and how many thereof were added to the sequence strand. As I was telling you, this pH change in pH after the addition will help you determine what and how many number of bases has been added to the sequence. This after that, this process, the DNTPs are washed away, and the process is repeated, cycling through different DNTP species. So for you remember what I said? Okay, if you add a DNTP, and what if there's a signal associated with it, if for example basically in this case a pH decrease, what will happen is, for each different species of the nucleotides present, of the bases present, ATGC, there will be a distinct change in pH, which will allow the ion sensor to identify what and how many of those bases has been added to the sequence. So this is how the how it will detect. So for T, maybe the pH change is high. For A also it is very high. For C and but depending on where it is, for this A it is quite low. For this A it has not been changed at all. So there will be very distinct signatures of these additions depending on the change in pH, which you can use to determine the sequence of the strand that is present. So just to give a recap what we are discussing. So next generation sequencing, we discussed pyrosequencing, which used light and luciferase enzyme to tell you what nucleotide is present at what position. Next, we talked about Illumina sequencing. This also used ddNTP sequencing by synthesis and also emitted light, and a machine captured and told you what base pair is present at that particular point. And SMRT, which basically also used a fluorescent signal and using different veils, and inside that there's a detector which will detect the fluorescent signal being emitted after a nucleotide has been added to the strand. And at last, we talked about ion Torrent sequencing, which basically uses the change in pH inside the medium to tell you what or how many of these nucleotides has been added to the sequence. Okay, coming to the last sequencing technology that we'll talk about is basically Oxford nanopore technology. So this is basically the latest that we have. That this is a very this is also a very widely used technology. So if you look at long read data sets, long read sequencing technologies. So this is one of the major long read sequencing technologies available here. Many you'll find if you go to Nature publications and you try to see the paper on paper in Nature Genetics, in many cases they've used this Oxford nanopore technology. So for one, sorry, yeah. So this is a very widely used technology that is there, and many papers will come across where they use either, if they're doing long read sequencing, either they'll use Oxford nanopore or they'll use PacBio SMRT. So these are the two major long read sequencing technologies available. By long read, I mean like the read length of the individual at one point of time, how much the sequencer can read, how many bases the sequencer can read. That is the read length. So the bigger the length, the better it is for you to understand and like detect variants inside the genome. We'll come to that a few classes later when we talk about how we detect variants, how we detect these big variations inside the genome. But long read sequencing is something that gives you much higher confidence than short read technologies like Illumina. So in this particular one, DNA is sequenced by threading it through a microscopic pore inside the membrane, and inside the membrane there's a protein that is present which unzips the DNA helix into two strands. This is basically the protein complex. So the upper protein unzips the DNA helix into two strands. So the whole DNA, one strand is left off, then another strand goes in. The second protein, this is the protein on below, creates a pore in the membrane and holds an adapter molecule. So this is the adapter molecule present, which basically holds the whole DNA fragment in one place. It does not fully pass through. And after that, what happens is a flow of ions through the pore creates a current, and each base blocks the flow to a different degree, altering the current. So what will happen is you are sending ions through. So here is your DNA, one single strand of DNA. What you're doing is you're sending ions through this whole protein. This whole you're sending ions through. So what will happen is it is creating a current in the flow of the direction of the flow of the ions. But since this is not empty, this has this DNA molecule. This has this DNA strand inside the protein. So what it'll do, it'll block the uh, flow of current. So depending upon the base present here, A, T, G, or C, the the, the blocking of this current will vary from base to base. So this is the output that you get, the variation of current, and each variation is a signature of what base it is present at that position. So what it does is basically it sends a current through the pore, and what it tries to check is how much of the current is getting blocked off. Depending on how much it getting blocked off, it will assign a particular base to that particular sequence. So it is studying how current is getting blocked off, and each of the bases have a distinct signature on how it can block the current. And as it is written here, each base blocks the flow to a different degree, altering the current. So depending upon how the current is, you can predict what your sequence is. So this is a much high technology sequencing thing that we have seen across all the sequencing stuff that we have seen. So we've mainly seen till now is sequencing by synthesis, but this actually does not do that. This does not use sequencing by synthesis. What it does is flows current through a pore in the same pore in which the DNA strand is present, and depending upon how the current is getting altered, it predicts a sequence. So apologies for the low resolution image, but this is all I could get. So the nanopore is inserted into an electrically resistant membrane. A potential is applied across the membrane so that the ions flow through the membrane, resulting in a current flowing only through the aperture at the nanopore. Single molecules that enter the nanopore, so one by one the strand is entering, the current is getting blocked in different manner. So the bases are so the strand is entering one base by one base. So first A enters, then T enters, then C enters, like this, one after another. The single molecules that enter the nanopore cause characteristic disruptions in the current, and measuring this by measuring this disruption, the molecule, what molecule is present at that position, or what molecule is passing through at that position, can be measured. So this is a technology that does not use any synthesis technique. So it does not synthesize a new strand or anything. It just measures how much a particular base is blocking the current flowing through a pore, and it can predict what base that is. So this is quite an exciting technology that we have till now. Uh, this is quite widely used, and like I should say that this is quite expensive if compared to a Illumina or a ion torrent sequencing. This is quite expensive than that. So this, although this is expensive, this is very accurate. So in case if you want to study long variations in the genome, these kind of technologies giving out very big reads. So this gives out around a, this can go up to a lot longer than what Illumina can give you. So if you want to detect those big variations inside your genome, this type of technologies are one that you should use. So pros and cons of the Oxford Nanopore technology. So pros involving minimum amount of data processing and complex algorithms, because you, in other cases, if you remember for Illumina, so for example here, what the is capturing is basically the base color is capturing is basically just the lights. It doesn't have any other information. So what it predicts is basically the probability of a particular base being at that position. But here, what happens is in Oxford Nanopore is basically you're directly inserting the DNA, and you're not synthesizing anything. You're just measuring what that particular molecule is, what particular nucleotide is blocking in terms of the amount of current that is giving you the base that is present there, which is much more simpler in terms of processing than to get the image of what it is going on, the whole image it is capturing, then it's predicting what bases are there, and that's not actually the sequence. The sequence is actually complementary to that. So a little bit of more of data processing is required for Illumina and other technologies compared to what you get for uh, Oxford Nanopore technologies like Oxford Nanopore. Uh, yeah, this can identify genomic sequences at large scales, as I was saying, because the read length is so large. The size of a pore is fixed and does not vary for large variations of the environment or experiment. Real-time sequencing at high rate of accuracy, it's 98%, but you have to consider, a lot of you will say this is not great because you've seen 99 and above accuracy in previous cases, but what you have to consider, the number of reads is very long, and that there is a trade-off between accuracy and the number of the length of the read. So if you are getting like a 10 times uh, bigger read with a 1% less accuracy, I think that is a good trade-off to have. Yeah, for using Oxford nanopore, it's about $1,000 that is required to sequence the entire genome, and the translocation of the polynucleotide through the pores are precise and in controlled manner. Now, the cons include uh, that the DNA sequence analyzing system is yet to reach its maximum efficiency because it's kind of a newer technology. Still algorithms are getting developed. Still, other stuff are getting developed for this particular technology. But yeah, there is still a long way to go. Uh, the handling the long strand of the single-stranded DNA is also a major concern. uh, what it can happen is so if your sequence has basically, say for example, you have this long sequence. At the top of your sequence there is a particular part of the sequence which is complementary to what it is present at the end of the sequence. So what can happen is once you pour the DNA inside the pore, the top part of the DNA can come back and bind to itself. So what will happen is it's self-complementary binding, which will hamper your ability to read the whole reads. One second. Yeah. Yeah. So controlling this is important because you do not want the complementary binding inside the pore because this will hamper your ability to read the whole sequence. And controlling the speed of ssDNA passing through the pore is yet to be achieved at maximum efficiency. It's still under a lot of work is being done on that as well. And high signal to noise ratio is yet to obtain. We do not have a high signal to noise ratio for Oxford nanopore technology. But keeping all the cons in mind, the, the only pro that you need to look at is the length of the read, because that is what will surpass all these cons. Because I am ready to have all these cons if I have a big enough read that I can confidently align to my genome and I can claim particular variants with high confidence. I am happy to have these kind of cons. So the read length is something that is very important. You will see that in future classes as well when you're trying to analyze what is different in your genome, analyze the variants, analyze the mutations, analyze the big variants. So these kind of stuff inside the genome, it's very hard to do with uh, short length of your read. The bigger your read, the better it is. So it's a trade-off, but I'll happily take a bigger read with these kind of cons. But as I said, it's a little bit more expensive than Illumina, so Illumina is still quite widely used in majority of the groups. So in my lab as well, we have an Illumina sequencer here. So we do not have a long read sequencer that is too expensive, but the Illumina sequencer is here. So it's just, Illumina is still quite widely used because of these technologies are still very expensive at this point of time. So, okay, that brings us to the end of this uh, the discussion part. I have summarized all the sequencing methods, how the read length and accuracy, and what are the applications and strength and weaknesses. If you want, you can just take a photo of this slide. This will help you in future if you want to like have a quick recap. So if you want, you can take a picture of this. But yeah, I'll not go through this. I've explained all of this in the last one and a half hours. So I just wanted to show you this slide. If you want, you can take a photo, or else you can move on to the assignment questions. If you have any questions, please feel free to ask in the chat box or unmute yourself and ask, please. Okay, then let's move on to the questions. So just to be clear about how we are doing this, so I'll keep the question on the screen for a minute or maybe a little bit less depending on the question. So it's mostly all are MCQ. So there are some multiple choices as well. So you can provide the answers in the chat box. I think that will be better than like just unmuting and speaking all of you together. So I'll keep the questions on the screen for about a minute. Then I'll check your answers and then I'll give the answers myself. And if there is any explanation needed to be done, I'll give it. Okay. So yeah, let's start. This is question number 1. Okay, let's see. Do we have any answers? Okay. The answer is A. That's correct. So DNTP will so DDNTP it's basically dideoxy. So there are two deoxy molecules there. So for normal DNTP, you won't have the just a 2' OH group. S- For DNTP, you will not have the 2' OH group. For DDNTP, you will not have the 2' OH group. Also, you will not have the 3' OH group as well. So yeah, the correct answer is A. So moving on. Let's see if we have an answer. Yeah, the answer is C. As we talked about, I'll just take you to the slide again. Yeah. So what happens is this is once so you have like adapters on the end, adapters on the top, and it crisscrosses and binds to the opposite adapters, and then it amplifies itself. So that is what is known as bridge amplification, and this is a signature of the Illumina sequencer. So yeah, it's true. Next. So somebody is trying to enter, I think. I give you another minute. Sorry guys. Okay. Yeah. I see a few people have given the answer D. I can understand how you are thinking. As in one way, it is true. But what happens is for small variations, small genetic variation in single nucleotides, what happens is if you have short read data, that is more useful to give you small information than long read data. I'll explain how. So, for example, you have a long read, and inside, say, you have 1,000 bases in one read. Inside the middle, say at 500th point, you have one variation, and the rest of the read is matching to the reference. So, what will happen is, since this is also an error-prone process, so we have a possibility of an error while aligning um, to the reads as well. So in bigger reads, the small variants will be missed out. So we'll consider them as an error and we'll miss them out. But with shorter reads, what will happen is there will be multiple of these shorter reads at that position, which will tell me on every read, however the small the read be, at every read at that position there is a variant, small variant, a single nucleotide change there. But in case of big reads, what will happen is you have big reads, so at one position your number of reads supporting that particular position will be lower. Getting me? So you have big reads, but the overlap between the big reads is lower compared to the overlaps in the small reads. So if you have small reads, if you have a single variant change, it will be much well supported in small reads, and it'll give you much more confidence to report in small reads than in case of large reads. But yeah, using LRS, you'll find papers, you'll find different kind of studies that has also identified small variations. That is doable. That is like LRS does quite fine, but the ideal way to go would be a short read technology like Illumina. So here the correct answer will be A and B. Uh, uh, D is not the correct answer. Okay. Next, moving on. This is an easy one. Let's see, do we have any answers? A lot of C's. The correct answer is C. Actually, yeah. This is the major reason why unique sequences are added to adapter ligation, so that you can find out from where in your original genome the read came from. Because the major factor is the genome is full of repeats. So if you don't know because a part of the genome here at say for example in chromosome 1 will be very similar to a part of a genome in chromosome 10. It can happen. I'm just giving it as an example, but it can happen. A part of the chromosome in position 1 is same to the part of the chromosome in position 2. So how will you differentiate a read coming from chromosome 1 and chromosome 10? You need to have a certain sort of marker. So that's why these unique index sequences are added to help you at least identify from where the read came from. Okay? So, okay, yeah, moving on. Let's see. The answer is both C and D. D is a lot of people were only saying C. C is obviously a correct answer, but strand displacement DNA synthesis is the way that you generate these hyphae reads, you generate those circular reads on both ends of the genome, and then you get the hyphae reads. So yeah, this both will be true. C is true, D is true as well. So, okay, moving on. Okay, let's see. We have any answers. Yeah, okay. Yeah, because it has the highest peak. This is very simple. Because the pH change is maximum, so that's the most added nucleotide according to the signals here. So yeah, it's quite simple. Okay, let's see what answers do we have. A few people actually said E. The relative signal intensity of light is proportional to the amount of base added and the amount of luciferase. It's not actually true. If I go back to the plot here, so what happens is this basically this ATP sulfurase helps in binding this PPI to the AMP to form the ATP, which basically binds to D-luciferin. So the presence of D-luciferin is important. The amount of D-luciferin present inside the cell is important because this is what will produce the luciferase enzyme. So the concentration of luciferase inside the well or the tube, it's not that important because it is getting formed from this D-luciferin enzyme. So the ATP will bind to this D-luciferin, which will form this complex. Now, since this complex is very unstable, it will like break off and give you the luciferase and the light. So yeah, it was a little trick question. So yeah, just to uh, just to clarify. And this APS, a few people might not know the full form. This APS, this APS is this molecule basically. Yeah, this ATP sulfurase enzyme, which basically is converting this AMP and the PPI released from this addition of this base to ATP. So, okay, okay, moving on. See, okay, Yeah, the answer is ABC. Somebody did not include B, I think, but B is the most important point of all. Uh, you're detecting how what bases are there. You're detecting the sequence by the changing keys. That's the whole principle of the ion Torrent sequencing technology. So please keep that in mind. Yeah, this is also quite simple. Yeah, yeah. The answer is D. That's the whole principle of it. It change. There's a pore. There's a protein with a pore inside it. The single-stranded DNA goes inside the pore one base pair at a time, and the amount of current that it is blocking off that emits a signature, which the signature is used to assign a base to that particular position. So yeah, okay. Let's see what else do we have. Okay. The answer is A. Some people are saying D. I just go back to the slide of uh, nanopore. So what essentially they are asking is this protein, and there is an adapter molecule that keeps the bases in place. But the protein above that is what helps unzip the DNA helix into two strands. So what they are asking is about this protein and with the adapter present inside that protein. So these are two different proteins present. Okay. So one protein present in top and the second protein present in bottom, which basically the second protein is the main one that is used for the sequencing purposes. The protein on the top is the one which unzips the DNA helix. So the question that was asked was about this protein, the protein that is helping in the synthesis. I agree it's a little bit ambiguous question, but yeah, sorry, but yeah, that is what they're asking. So the correct answer will be A. It directs the DNA strand properly through the pore because it has that adapter molecule present inside itself. So that is the motor protein present on in the below. Okay. Okay. Regarding PPT sharing, see, I am supposed to submit this PPT to the NPTEL folks. Uh, I think you have to ask it from them. I am not supposed to give you this PPT directly to you. But what I do is basically I have to upload the whole lecture for these 2 hours. I'll have to upload it on YouTube. So you can go to YouTube and search for it. You'll find the whole video. But I cannot directly give you the PPT because I am supposed to give the PPT to the NPTEL folks. You can ask it from them. If they give it to you, it's you can get it from them. But I am not supposed to give it directly to you. So yeah, okay. Coming back to the questions. Yeah, the answer is B. The main function of luciferase is using the ATP as a substrate and converting the luciferin to oxyluciferin, which is from the light. So, also regarding the PPT, what I'll do is I'll ask the NPTEL people if they're happy with if they give me permission. So what I do is I have to upload the PPTs in a Google Drive and share the link with the NPTEL people so that I can share that link with you as well. But first I have to take the permission from them. So let me ask it after this class. So from next class, if they say I can share my PPTs, I'll give you all the Drive link where you can see the PPTs for all the weeks. Like I have not uploaded it all the weeks all together. So after the class for each week, you'll get to see the PPTs. But first, I have to ask for permission. So I'll be able to tell you next week. Coming to the answers. Okay, how to search on YouTube? You just search my name. My channel will come up. This is my name. My channel will come up, and in that channel, this video will be uploaded. So, okay. Let's see the answers. Okay, actually, I'll give you the link. Just a second. I'll give you the link to my channel. Okay. Uh, let's check the answers. The answers is A and C. The first issue with Illumina is the read is short. That's the major drawback of Illumina. Illumina gives you around 150 to 300 base pairs, which is quite short. And since it is a short read, as I was saying, if you have say you have a repeat region where the number of repeats, say for example, ATGC, and it is repeated ATGC, 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 like that it is repeated, say for 500 times. So 500 into 4 ATGC, that will be like total 2000 basis range of repeats. It is a duplication of 2000 length. So if you have a read of 150 base pair length along this 2000 base pairs, where will you align the 150 base pair? Because everything is repeat. So what happens is for repeats, this short read thing becomes an issue because in most cases the number of repeats far exceeds the number of reads, the length of the reads that you are getting from short read data. So that is a major issue with Illumina as well. So if you want to, this is a major thing that we face if you want to study duplications, big duplications inside the genome. One such example is Huntington disease. If you want, you can just search it. It's a duplication region. So it's a smaller duplication, but still, Illumina using Illumina, it is very hard to align those particular reads to that particular duplicated region. So that becomes an issue. So A and C are the correct answers, and yeah, moving on. A fairly simple one, I think. Yeah, I agree. Pushpa, paired end sequences can solve the problem of repetitive genome, but that also has to be within a certain range. Na? Like if your read is 150 base pair range, you can read 150 base pairs from here, 150 from here. So total 300 you can read. Up, out of that, you don't know, na? Because like you're reading 150 from here, 150 from here, so 300 you are at least confident about that too if you are taking no overlaps. So it's 150 or 150 ekdam, like one ends the other starts. So that is the extreme case. Even in the extreme case, the maximum you're getting is around 300 to 400 base pairs, not more than that, right? But the repeats can be like 1000 base pairs long, 2000 base pairs long. How will you resolve those repeats? You don't know where, because along the repeat, the sequence is same. The sequence is same for 1000 base pair range. You cannot claim that a 150 base pair read from your sequencer is attaching at say the 50th position of the 2000 repeat range. You cannot say that it can attach to the 50, it can attach to the 500, it can attach to the 1000, because it's only 300 from both sides, right? So even though paired edge sequencing can solve the problem of a repetitive genome in certain cases, but in most bigger repeats, it's of no use because the repeat region is very big. You need big enough reads to kind of confidently say that this read aligns to that repeat region. Otherwise, it's become very difficult. So one of the reasons it's like if you follow genomics, so the whole Y chromosome is a uh, very repetitive genomic region. The whole Y chromosome, it has a lot of repeats. So just last year only, like in 2023, the whole Y chromosome came out. So this was the paper, the complete Y chromosome. This is a 2023 paper. So until 2023, I'll give this paper link here. Until 2023, we did not know the sequence of the whole Y chromosome. And this is one of the reasons, because with short read technologies, was very hard to get the whole Y region. Now with long read, now with other types of data that we have, multimodal data sets that we have, we are able to resolve this YDPs. So go through this paper, you'll like it a lot. This is a 2023 paper, and like, it last year came out, and at till 2023 we did not have a genomic reference with all the Y chromosome together. This is the paper after which the T2T this is basically the T2T consortium paper. This came out after which the whole Y chromosome has been resolved, and now we can confidently have reference genomes with Y chromosomes in it. So yeah, read it, you'll like it. And regarding the question, yeah, it's quite simple. It's the microarray doesn't do sequencing by synthesis. It has limited ability to detect structural variations and novel mutations. Novel mutations, why? Intuitively thinking, because you need to have DNA probes inside that, basically the slide, you need to have DNA probes with known mutations that you expect from the cell. That is how if the known mutation is present inside the cell, that will come and bind like this. But if you don't know what kind of mutations are present, and specifically structural variations, like they can be like 500 to 1000 base pair long, so those big variations, you don't know uh, uh, how to kind of measure those variations using uh, microarray. So keep this in mind. Microarray only works if you have a good amount of knowledge about how the genome is looking, what kind of mutations to expect, and everything. And obviously, it is cost effective, and yeah, it relies on probes. Okay, that brings the end of our session. So I start session at 6. A lot of people joined later. I'm not sure if the timing has been communicated wrong to you, but this today's class was from 6 to 8. In next week, my timing has been shifted. It has been preponed an hour, so I'll be taking from 5 to 7. So please join if you want, and if you are not able to join, please go to after the class. You can see this videos being uploaded on YouTube. You can go and take a look there. Okay. If nobody has anything to ask or say, I'll close the session here then. Okay, guys. Thank you for joining. Thank you for attending the class. You guys have missed the first part of the class because many of you joined late. If you want, you can go check out the video. I'll upload it in after this, so maybe in an hour or two, you can go and check out the video. Otherwise, yeah. Thank you guys. Thank you for joining. See you next week.